Digestion in buccal cavity. Saliva contains 98% water and 2% other constituent like electrolyte, sodium, potassium, calcium, chloride, bicarbonate, and digestive enzyme salivary amylase. The only chemical digestion that takes place in mouth is by the action of salivary amylase. Salivary amylase helps in conversion of starch into maltose. About 30% starch gets converted into maltose in mouth. Maltose is a disaccharide. Saliva also contains lysozyme. It acts as an antibacterial agent that prevents infection. The food here is called as bolus. The bolus further passes down through the esophagus by peristalsis. Sometimes regurgitation or vomiting takes place due to reverse spasmodic peristalsis. Food from the esophagus enters the stomach. The gastroesophageal sphincter controls the passage of food into the stomach. Digestion in the stomach. Stomach stores the food for 4 to 5 hours. The physical digestion happens by churning of food. Thick muscular wall of stomach helps churning process. Churning further breaks down the food particles and also helps in thorough mixing of gastric juice with food. The mucosa layer of stomach has gastric glands. Each gastric gland has 3 major type of cell, namely mucus cell, peptic or chip cell, and parietal or oxyntic cell. Mucus cell secretes mucus. Peptic cell secretes proenzyme pepsinogen. Parietal cell secretes HCl, an intrinsic factor which is essential for absorption of vitamin B12. The gastric juice contains mucus, inactive enzyme pepsinogen, HCl, and intrinsic factor. In infant, stomach also secretes renin. Mucus protect the inner lining of stomach from HCl present in gastric juice. HCl in gastric juice makes the food acidic and stops the action of salivary amylase. It kills the germs that might be present in the food. Pepsinogen gets converted into active enzyme pepsin in the acidic medium provided by HCl. In presence of pepsin, proteins in the food get converted into simpler form like peptones and proteose. Renin found in gastric juice of infant act on casein, a protein present in milk. It brings about curdling of milk protein with the help of calcium. The coagulated milk protein is further digested with the help of pepsin. Renin is absent in adult. At the end of gastric digestion, food is converted to a semi-fluid acidic mass of partially digested food is called chyme. A chyme from stomach is pushed in the small intestine through pleuric sphincter for further digestion. Digestion in the small intestine. In the small intestine, intestinal juice, bile juice, and pancreatic juice are mixed with food. Peristalsis movement of muscularis layer helps in mixing of digestive juices with chyme. Bile juice and pancreatic juice are poured in duodenum through hepato-pancreatic duct. Bile juice is dark green colored fluid that contains bile pigment, bilirubin, and biliverdin. Bile salt, sodium glycocholate and sodium taurocholate, cholesterol and phospholipid. Bile does not contain any digestive enzyme. Bile salt neutralize the acidity of chyme and make it alkaline. It brings about emulsification of fats. It also activates lipid digestive enzyme or lipase. Bile pigment impart color to fecal matter. Pancreatic juice secreted by pancreas contains pancreatic amylases, lipases, an inactive enzyme trypsinogen, and chymotrypsinogen. Pancreatic juice also contains nucleases, the enzyme that digests nucleic acid. The intestinal mucosa secretes digestive enzyme. The goblet cell of mucosa produce mucus. Mucus plus intestinal enzyme together constitute intestinal juice or succus entericus. The intestinal juice contains various enzymes like dipeptidases, lipases, disaccharidases. Both pancreatic and intestinal lipases initially converts fats into fatty acid and diglyceride. Diglyceride are further converted to monoglyceride by removal of fatty acid from glycerol. The mucus and bicarbonates present in pancreatic juice protects the intestinal mucosa and further provide alkaline medium for enzymatic action. Submucosal Brenner's gland helps in the action of goblet cell. Most of the digestion gets over in small intestine. Let us study the action of pancreatic and intestinal secretion in sequential manner. Action of pancreatic juice. Pancreatic amylase acts on glycogen and starch and convert those to disaccharide. Lipases hydrolyze fats molecules into fatty acid and monoglyceride. Inactive trypsinogen present in pancreatic juice is converted to its active form trypsin. This conversion is brought about by angiokinase present in intestinal juice. Trypsin converts protein as well as proteoses and peptones to polypeptides. It also converts chymotrypsinogen to active chymotrypsin, and chymotrypsin converts polypeptides to dipeptides. Nucleases present in pancreatic juice helps in digestion of nucleic acid to pentose sugar and nitrogenous base. Means your nucleic acid by the action of nucleases get broken down to nucleotides. Nucleotides are broken down to nucleoside, and phosphate enzyme here is nucleotidases. Nucleosides are further broken down to sugar and nitrogen base. The enzyme here is nucleosidases. Here conversion of proteins into amino acid, fats to fatty acid and monoglyceride, nucleic acid to sugar and nitrogenous base, and carbohydrates to monosaccharide marks the end of digestion of food. Food is now called cell. Cell is an alkaline slurry which contains various nutrients ready for absorption. The nutrients are absorbed and undigested remains are transported to large intestine. Remember, mucosa of large intestine produces mucus but no enzyme. Some carbohydrates and proteins do enter the large intestine. These are digested by the action of bacteria that live in the large intestine. Carbohydrates are fermented by bacterial action, and hydrogen, carbon dioxide, and methane gas are produced in colon. Protein digestion in large intestine ends up into production of substances like indole, skatol, and H2S. These are the reason for odor of fecus. This bacteria synthesize several vitamins like vitamin B and vitamin K.